He earned his bachelor degree from Colby College, followed by PhD in MIT. This young, handsome guy without a beard is Bill. He was a model for Norman Rockwell's famous painting called Right to Know. And he moved to Europe for 2 courses of postdoc trainings. And he came, in 1982, he came back to the United States to run his own lab in Johns Hopkins. And in 1996, he moved to UK for a new position as a professor and principal research fellow of Wellcome Center for Cell Biology and the University of Edinburgh. And he still continues his position now. During his academic career, he achieved really a lot. I cannot summarize everything, but what I can say is that he discovered most of the major proteins for mitotic chromosome formation that we know of so far and found how they work. Most of them are now the basic knowledge in cell biology that you might have learned from your undergrad. He published nearly 400 research articles, and around 20 of which are Nature Science Cell. Not nature something, it's an actual nature. And for those countless achievements, he was not shy to adapt the most cutting-edge technologies like mass spec, machine learning, high-C, and now even polymer modeling. And he also wrote an excellent cell biology textbook, which is now widely used throughout USA and Europe. Now, today, I was asked by many people how I know Bill. To explain that, I have to introduce my PhD supervisor, Dr. Damien Hudson. Damien was a postdoc at Bill's lab for nearly 10 years in Edinburgh and came back to Melbourne University, and I was his second PhD student. Although I am a professor now here who pretends to know everything, but one day I was very young and didn't know anything about science. But Damien taught me everything from the basic, like teaching a baby how to walk, how to eat, and how to drink. Basically, he was like the father of my research career. Unfortunately, he passed away by cancer at young age, just 2 years before I came to KAIST. But anyway, because of that relationship, one day Bill generously invited me to his lab, and Damien said, let's complete our ChIP-seq protocol in his lab. And I spent 6 months in Bill's lab in 2010, and afterward, we continued our good relationship. Every moment that I had to make a life-changing decision, I always asked Bill, should I go to UPenn? Should I accept the GSK offer? How should I prepare the application for a faculty position? And everything. He gave me advice. I listened, and it always worked. If Damien was like the father of my research career, Bill is like my grandfather and is an amazing mentor of my life. Now, it is the moment that I have dreamed for a very long time, which is to invite Bill to my school where I serve as a faculty member and introduce you guys. Today, he will be introducing an exciting result from the chromosome formation team, each member of which is the world leader. Now, it's you, Bill. You have not taken my mic.
So there's Walter Fleming, and he named mitosis because he saw this cell nucleus turn into what the Greek word mitos means threads, and he saw these threads form inside the cell nucleus. So he decided to call it mitosis. And the product of that formation of chromosomes in humans is here is the human karyotype done, but this is the RPE1 cell line. So, so those are one of the many ways that mitotic chromosomes look, but there's a big problem here. So how can you possibly fit all that DNA into such a small object as this mitotic chromosome? That's the question. How do you fit it in? And my friend Frank Ullman made this slide, which I think maybe is a little easier for some of you to understand. So consider the London Underground, which is 402 kilometers. Now put it all into a small suitcase. And then that's what you need to do to make a mitotic chromosome. 10,000 times shorter. Whoop. Oh, 10,000 times shorter. Okay. So how are these chromosomes put together? Well, maybe they're coiled, you can hear me, yeah? Maybe they're coiled helically. That was first reported all the way back in 1880 by Bernetsky. And you can see very clear, helical coils. Absolutely true. They must be helically coiled. Oh, no. No, maybe they're not helically coiled. Maybe it's just like a plate of spaghetti. And that was proposed by Ernest Duprat in the 1960s, and then Kazuma Yoshima and Mikhail Eltsov from very sophisticated cryo-electron microscopy in 2012. So maybe that's it. No, maybe not. Maybe they're just loops. And that's what Uli Lemley proposed back in 1977. They can't hear me? Are the Zoom people telling you they can't hear me? Okay. I'm sorry. So the Zoom people are going to hear me, and you're going to hear me all distorted and loud. I would rather make the Zoom people suffer. But so if we put chromosomes in this very low ionic strength buffer, they swell up. And they swell up, and if we then centrifuge them onto an electron microscope grid, they look like a big pancake. All right. Now what we're going to do is we're going to take that grid and we're going to dip it into uranyl acetate, which is a stain, but it's also a charged molecule. And what happens is most of the chromatin goes back, but what you can see on the surface of the grid is loops. So this chromosome obviously must be organized with many, many, many loops of chromatin, and the average size of these loops is about 80,000 base pairs. Well, if you have a loop, how do you make the loop? Something has to hold the bottom of it. I'm not going to hold that microphone. I'll take the Coke. I'd rather have the Coke than the microphone. So Uli Lemley suggested that to make a loop, you have to hold on to the bottom. So he suggested that there might be a protein scaffold that organizes these loops. And that was big paper in Cell, very exciting. But then there was a huge fight. And so this was the story of my life for a long time. There were vicious fights about whether this scaffold that he saw was real or it was an artifact. And so I decided that the way to tell if it was real or not was to isolate the scaffold proteins, make antibodies to them, and look at them and see if they were there. People had said that the way you make a scaffold, I'll show you. Well, actually, I'll show you now, maybe. The way you make a scaffold is you take mitotic cells, you isolate the chromosomes, then you chew up the DNA, and you solubilize away most of the DNA and most of the proteins. And when you do that, they're not gone. There's still something that looks like a chromosome. But this is not, there's only about 2 or 3% of the DNA is left here. This is mostly protein. But people said, no, that's just precipitation. It doesn't mean anything. That was the argument. So what I did was I isolated these and I made scaffolds. And then I ran them on a gel. Now, this all sounds very easy to you, but I should just take a minute and go much over my time and tell some stories. So, for example, this was 6 months worth of work, which I ran on one gel. 6 months worth of isolating chromosomes and making scaffolds, putting them all in one sample and running them on 2 gels. And I sat and I watched those 2 gels run in case something went wrong. Then I cut the band out of the gel, and I injected it into a guinea pig. Because I didn't have enough antigen to inject a rabbit, I injected it into a guinea pig, but I just had a bad feeling. So I took a can of red spray paint, and instead of writing my name on the wall, I put a dot on the back of the head of the guinea pig. And then I went back 1 month later to boost my guinea pig to make the antibodies, and every guinea pig in my cage was white. So I walked around in the animal room, and in somebody else's cage, there was a guinea pig with a red dot on the back of his head. So I said, I'll have you. That's just true. And that guinea pig made that antibody. And, well, the question is, what was the protein? Well, I had a friend who worked on the enzyme topoisomerase 2. And what we discovered was if you took our antibody and stained his topoisomerase 2, you saw a band, and you saw a peptide digest, natural peptide digest. So that's his antibody to topo 2. That's my antibody to our scaffold protein. It's the same protein. It's a perfect experiment. It's topoisomerase 2. So that was the first chromosome scaffold protein that was identified. So the question was Lumley right, and was it at the base of the loops? So we decided to look at a mitotic cell. So this is actually a big picture of one cell, which has been swollen in a hypotonic buffer and centrifuged and put on a glass slide. And now let's turn on the channel with the topoisomerase 2. And there it is. It's at the bottom of the loops of DNA. So yes, topoisomerase 2 was right where the scaffold proteins were supposed to be. So that fit with the idea. But that was a very difficult, I can't spend 6 months to identify every protein. We needed to do something a little bit more efficient than that. So what we did was we isolated chromosomes through this rather simple looking but actually quite difficult procedure. We got chromosomes and we could see many bands on a gel, and then we put them into a mass spectrometer. And this was done in collaboration, was done by my postdoc Shinya Ota in collaboration with Jimmy Carlo in the laboratory of Yuri Rapsilber. And we identified over 4,000 proteins that were in the chromosomes. And then, actually, this is a very nice story, but it's not a story for today. We had to use machine learning to figure out which of those proteins were actually in chromosomes. Because actually, when the nuclear envelope breaks in mitosis in human cells or invertebrate cells, the chromosomes form, the nuclear envelope breaks down. And then what happens is a whole bunch of cytoplasmic proteins just stick to the chromatin. And you can't purify them away from it. So how do you tell what's really chromosomal? So that was machine learning. And so, of course, most of the chromosome is the histones because they're in the nucleosomes, but they're not in the scaffold. We get rid of them. So what is in the scaffold, amongst other proteins, is a group of proteins called SMC proteins. Now, those proteins were discovered in Russia, in Vladimir Larionov's lab, in yeast genetics, and SMC stood for stability of many chromosomes because that was their asset. But in fact, his postdoc left Russia, moved to the United States, and decided to call them structural maintenance of chromosomes instead of, which is a better name anyway. So these proteins are called SMC proteins, and they are critical. So SMC proteins organize the behavior of mitotic chromosomes. So, for example, a complex of SMC proteins called condensin, that's what's responsible, I'll show you today, for shaping mitotic chromosomes. And another complex called cohesin is what holds the replicated DNA molecules together so that when the cell divides, each replicated DNA molecule splits so that one goes to each daughter cell. And this cohesin complex is also very important in organizing loops in interphase. And so mutations in cohesin proteins cause Cornelia de Lange syndrome. It's a developmental disorder and other disorders. So there are many people who study gene expression who study SMC proteins, but there are also cohesin, but there are also people who study chromosomes. Now for the bad news for those of you who don't know this. There are two condensin complexes. That's not bad news, but the bad news is they're named in the order in which they're discovered. Well, it kind of makes sense. The first one you discovered you call condensin 1, the second one you call condensin 2, but condensin 2 acts first. Condensin 2 is in the nucleus, and it acts first. Condensin 1 is in the cytoplasm, and it acts second. So this is not very helpful for you if you don't know about this, and it's not my fault. Okay. We discovered one of these proteins, but we didn't name these complexes. So the way these SMC proteins work is by extruding loops. Okay, now, Jihoon, help me out, because this is a video that should play without me having to do anything. There we go. All right. On the left, we're going to see a condensing complex binding to DNA and extruding a loop. And on the right, we're going to see a simulation done of what happens if you have many loops extruding with the average lifetime of these loops on decondensed DNA molecules. So the condensing, that's what's meant by extruding a loop. And then if we watch this video on the right, you will see that actually what happens is just by extruding loops and by entropic exclusion of those loops, you end up forming rod-like molecules, rod-like structures. All right, so this is loop extrusion, and these are both from the work of Anton Golobrodko when he was a PhD student with Leonid Mirny, who was one of our collaborators. All right, so that top band was topoisomerase 2. That band turned out to be SMC, a protein called SMC2. And SMC2 is that protein. So there are two condensing complexes, but the way they work is that they both have SMC2 and SMC4. They have two different sets of these guys. So if we get rid of SMC2, there's no condensin, neither complex. They're both gone. So that's what we're going to do. We're going to get rid of SMC2, and the condensing complex is going to fall apart, and we're going to ask, what does this do to the structure of chromosomes? Now, the way we're going to do that is to use a technique called serial block face scanning electron microscopy, which probably most of you don't know about, so I'll tell you. So what we're going to do is we're going to prepare a specimen as though we were going to do traditional electron microscopy, which is that we're going to take cells, we're going to fix them with glutaraldehyde, we're going to do correlative microscopy, which means at light microscope level, we're going to look on a slide and we're going to identify a cell that we want. Then we're going to embed the sample in plastic, and we're going to find that one cell, position it in the center of a plastic block, and we're going to mount it in this special microscope called a 3-view microscope. So there we have a specimen in a block. This doesn't look like a microscope. That looks like some kind of blade, and it is. So what's going to happen in this microscope is we're going to lift up the specimen. We're going to take the knife, and we're going to cut 60 nanometers off the top of the plastic specimen. Oh, shit, okay. What was it you did, Ji-Hun? We're going to retract the knife, and then so this is the opposite of what if you know about electron microscopy, this is the opposite of what you think you do. Usually for a classical electron microscopy, you cut a thin section and you look at the section. No, we cut a thin section, we throw the section away, and we look at the block. This means instead of having to get 300 sections, which are all perfect, no, we just see all 300 slices of the block. So here's a DT40 cell from top to bottom, 300 sections. So this would be incredibly hard to do by traditional electron microscopy, but by this method, it just takes a week. All right, so now we're going to do this to a cell where we've gotten rid of the condensin, so there's no condensin. So the first thing we do is we section. And then using a very sophisticated image processing protein, we do what's called modeling. So that is, we recognize the chromatin away from the rest of the cell. And then we do segmentation, which is we separate the separate chromosomes. And then we can analyze it. But what you can see is this doesn't look like we got rid of condensin, and this doesn't look like mitotic chromosomes anymore. So a less fancy way of doing that is just to look in the light microscope, and on the top, we see cells entering mitosis. And here we see cells entering mitosis under the same time scale, but without condensin. And this looks like an interphase nucleus. It doesn't look like mitotic chromosomes, but look how big it is and how big that is. And remember that volume goes with the cube of the radius. So this is highly condensed. This is much smaller in volume than that. So the chromatin is highly condensed, but it does not form chromosomes. So you require condensin in order to form chromosomes. Right. So here are the first set of our conclusions. The chromosomes are organized by a scaffold of non-histone proteins. It's a stupid name. It just means they're not histones, but nobody knew what they were, so they called them that. There are SMC complexes that work by loop extruding, as you saw in that movie, and they're key components of that scaffold. So cohesin organizes the interphase genome, and it's published widely in the literature that it comes off in mitosis. Condensin 2 was in the nucleus, and right from the very beginning of mitosis during prophase, condensin 2 is organizing the chromosome into these loops. Condensin 1 is in the cytoplasm, and later on, when the nuclear envelope breaks down, it starts acting and helping to condense the chromosomes. In cells rapidly depleted of condensin, the chromatin is compacted, but chromosomes are not shaped correctly. So mitotic chromosome formation involves two things that are operating in parallel. One, we're making these shapes that look like chromosomes, and the second one is we're compacting the chromatin, and those two processes can be separated. So now we'll talk about some of our work on chromosome formation. So the main problem of working on mitosis is that you want to study mitosis, but you look at HeLa cells, which are a rapidly growing cultured cell line, and 6 to 8% of the cells are in mitosis. How am I going to study them? Well, I work with a fantastic woman, Kumiko Samajima. She's been working with me for 20 years, and she is amazing. She's a genius. She's the best molecular biologist I know. She's an incredible geneticist. She's a biochemist. She does super resolution microscopy. Kumiko can do anything. And she runs marathons. She and 3 friends ran across Scotland in 26 hours. That involved 2 marathons worth of running. So one day she came into group meeting and said, I found something very interesting. And she found a source of a chemical genetic way to make mitotic cells any time we want. So this is the cell cycle, very, very simplified. And as I think many of you know, the cell cycle is run by cyclin-dependent kinases and their cyclins. And the one we care about is CDK1. It's the sort of, well, it was the first one identified, and it's a master regulator of mitosis. So what we are going to do is we're going to replace CDK1 with an analog-sensitive allele. So this means we have made a mutation in the ATP binding pocket of CDK1 that allows this inhibitor to fit in. This inhibitor is bigger than ATP, so it will not bind to any other kinase in the cell, but when it binds to this analog-sensitive CDK1, it inhibits it because the kinase can't use ATP anymore. And what happens if you inhibit CDK1 is the cells all accumulate at the boundary between G2 and mitosis. And then another nice thing, which is not true of every inhibitor, is if we now wash the culture, the one in MPP1 comes right out, and the cells enter mitosis synchronously. So the allele we're using Xenopus CDK1 in a chicken cell line. Doesn't matter, Paul Nurse got the Nobel Prize partly for showing that human CDK1 would drive the cell cycle in a yeast. So they're not species specific. And it was made by a guy named Helfred Hogeker, who's another friend. So my PhD student, Jan Rupert, made this movie for me. So these are HeLa cells. And you probably know, I just told you that if we study HeLa cells, we have 6 to 8% of the cells in mitosis. I mean, how can we really do biochemistry with that? So let's wash out the one in MPP1. Now they all go into mitosis, or almost all of them. Right, so we can get 96% prophase cells, which normally you would find 0.5%. And we get 98% cells in later stages of mitosis. So now we can make cells enter mitosis whenever we want. So what happens to the SMC proteins during mitotic entry? So we're gonna do this protocol where we're going to arrest the cells, we're going to release them into mitosis, and then at very short intervals afterwards, we're going to let them off, and we're going to ask, in the first case, what proteins are on the DNA and what proteins come off. And we're going to do it using a method called chromatin enrichment for proteomics, developed by my colleague Georg Kustacher. So we're going to cross-link cells with formaldehyde, just like you would do for a chip experiment. If you work on epigenetics, you know what chip is. We're going to lyse the cells in detergent. We're going to spin down the nuclei. We're going to digest with RNase. We're going to solubilize the nuclei with SDS. And we're going to mix with 8 molar urea and spin. So what's happened is proteins on the DNA have been cross-linked to the DNA with formaldehyde, and even though these conditions are all very strong denaturing conditions, those proteins stay stuck on the DNA. And so we spin, we wash this pellet, now we get rid of the DNA because we're not interested in the DNA, and we look at the proteins. And we do that by mass spectrometry again. So what CHEP does is identifies that are proteins that are close enough to DNA in order to cross-link to it. Now, you wouldn't think that this would be a political statement, but it is a political statement, as you will see. So this is the map of the over 2,000 proteins that we identified in this method. And we're going to use t-SNE, which is t-distributed stochastic neighbor embedding, to map them. This map is rather arbitrary, but it depends on how you set it up. Certainly the dimensions in the map are arbitrary, but what t-SNE does, it's a way of taking a multi-dimensional data set and taking things that behave similarly and trying to put them close together. So that's what a t-SNE map is. But this is, as you can look at this and you remember where I live, this is actually a political statement because we call this our UK map. Okay? And furthermore, during mitotic entry, Scotland is on the way up, and England is on the way down. And I'm a member of the Scottish National Party, so, okay. So it's a political statement. All right, so here's that map, and there's cohesin. So cohesin tends to come, it's in southeast England, so that means cohesin is very rich. And also, it comes off during mitosis. And condensin is in Scotland, and it tends to go on during mitosis. So cohesin drops shortly. Condensin 1 accumulates after nuclear envelope breakdown. Condensin 2, actually, I didn't show. The specific condensin 2 is around in here. But quantitative proteomics, so this is what everybody believes about how condensin and cohesin work. But our proteomics data actually gave a big surprise. It's not going to be a big surprise to you if you're not excited about this, but it's a big surprise to us. So this is a marker that Kamiko Samajima made. So this is it's 4 times GFP hooked up to a nuclear export sequence. So this lives in the cytoplasm. And so there's the empty nucleus. And at this point is when the nuclear envelope breaks down. This is 3-minute time points going into mitosis. And because the synchrony we have with these experiments is so perfect, we know exactly when the cells went into mitosis. And so this is when the nuclear envelope starts to break down. But actually, if you look in the middle, you can see proteins from the cytoplasm are coming in before. And so here you can see that. So proteins from the cytoplasm are coming in before the nuclear envelope breaks down. That's a surprise to many people. But then if we look at the behavior of the molecules I've been talking about, condensin 1, well, remember, condensin 2, that's the one that lives in the nucleus. So it's there in the nucleus. And actually, over the course of mitosis, a little bit of it comes off of the chromosomes. Cohesin drops quite a lot as cells go into mitosis. And everybody says that cohesin is gone in mitosis. And then condensin 1 is the protein that's in the cytoplasm, and it increases a lot. And this is quite important that it does this, and you'll see why in a minute. But this is by normalizing the amounts that we've detected in the mass spec to what's there in G2. What if we take the absolute numbers and score how much of these molecules is there? Then we get something that, when this paper is submitted, is going to be we're going to have a bad time with referees. Because there's a ton of this cohesin molecule. So even though the levels drop, they only drop down to about the level of the molecules that are supposed to be making the chromosomes. So there's still quite a lot of these cohesin molecules on mitotic chromosomes. So the first study, which I'm going to summarize for you in the beginning, we only studied condensin because only condensin is supposed to be involved in making chromosomes. In the last work I'll talk about, at the end of the talk, we asked about what does cohesin do? And cohesin does a lot. It's not supposed to, but it does. So where have we gotten? We developed this system, or Kimiko. I didn't know anything about it. I just sat there and my jaw went, ugh, when she showed the first picture. She showed a slide where all the cells were in prophase. I'd never seen anything like that. Okay. Kimiko did that. So using the analysis of that, we showed that chromatin proteins undergo a very reproducible program of changes during mitotic injury. I didn't show you the data, but the first thing that happens when cells go into mitosis is they change RNA protein interactions, not chromatin protein interactions. It's very surprising. Anyway, there's a paper in 22 in Molecular Cell. You can read about that. Cohesin and condensin SMC complexes behave very differently. Cohesin levels dropped dramatically, but there was so much there that there's still a lot as the chromosomes form their rod-like shape. There's just as much cohesin as there is condensin in early prometaphase. Later on, the cohesin's all gone. So that suggests that cohesin might be doing something to help shape mitotic chromosomes. So also, I didn't show you the data, but in these cells, mitotic chromosomes form on the inner surface of the nuclear envelope. And that's going to be very interesting. So I'm going to show you some polymer modeling that's been done, and they haven't done polymer modeling on a surface yet for these. So that's going to be quite interesting. Okay. So now I'm going to go to our work that we did looking at high C and modeling. So to figure out what condensin was doing, I don't know if all of you know about high C, so I thought I would introduce high C. You may know that high C is an American fruit drink. Europeans have no idea why the name high C is funny, because that drink doesn't exist in Europe. So basically, when you do CHIP or you treat nuclei with formaldehyde, you cross-link proteins to DNA. So you handcuff DNA molecules that are close to each other by cross-linking them in a mess of protein. These don't have to be specific protein molecules. It's just trapping the DNA. Then the next thing that we do is we cut this with a restriction enzyme or with another enzyme, and then we fill in the end overhangs, and we put biotin there. And then we dilute things, and then we just do a ligation. So what we tend to do is we tend to ligate molecules that are trapped together by being held in with this cross-link to protein. Then we shear the DNA, and then we use the biotin, and we purify the ends. And then we sequence millions and billions of base pairs. I think Joop Dekker's sequencing bill at one point for this project and other projects was $100,000 a month. And Howard Hughes said, no. So the data that you get are heat maps. And so many of you have seen a high C heat map. And just for those of you who don't know what this means, this is how it works. This is how we think about it. So what we're doing is we're taking the sequences in one chromosome here, for example, and we're starting at one end and we're going to the other end, and then we're starting at one end and going to the other end. And whenever we have a hit, so whenever we've linked two things together, we have a spot on the map. So what happens if I have a spot there? Well, if I have a spot there, region 10 on the chromosome has been cross-linked together with region 7 on the chromosome. And so if you think about what that means there must be some kind of a loop. So 7 is close to 10. Okay? Now, if you know how to think about this in the right way computationally, you can take this map or plots that are derived from this map, and you can make a structure, a predicted structure for the organization of the chromatin in the nucleus. So we published a paper about this in 2018, and I'll summarize that. So at the beginning of mitosis, one of the things that we saw, or what we saw in the high C map, was this kind of not very interesting line. And this is called a scaling plot. And this is 10 kb out to 1 gigabase here. So it's a plot across the size range in the chromosome, and this shows how likely sequences are to be cross-linked to one another. And so if we took this plot and we analyze the data going away from that line, it's like analyzing the data going along on this line. And this means that for the features that we see here, we can give them an absolute size. We know how big they are because that's an absolute scale. And the only interpretation, the polymer interpretation of this structure that works, is that what's happened to the genome in the beginning of mitosis is just got turned into a chain of loops. So in interphase nuclease, it was organized into heterochromatin and euchromatin. In mitosis, it's just a chain of loops. And in the beginning, these loops are, because we know the size, these loops are about 60,000 base pairs. But that size changes, and that's not important. Okay, so that's what we saw in the beginning. But we saw something later. So later on, and this was one of those so the way collaborations, big complicated collaborations work like this is two PIs meet at a meeting. One PI sees that the other PI has something they want. They try to figure out a way to convince them to give it to them, and they set up a collaboration. And then the first PI goes home and tells the people in his lab, please make the sample. And the PI in the other lab goes home and says, please analyze the sample. And so the person makes the sample, and they send it to the other lab, and the other person goes, I'm going to analyze the sample. And so it sits there for like 2 years. And then somebody runs an experiment, and then you get an email saying, I can't believe this. What's going on? Because what happened was nobody had ever seen anything like this in a high C map. You not only had the middle line, but you had these other lines that appeared on either side of it. And what do they mean? So the lines tell you that sequences are close together. What this map is telling me is that every sequence on the arm of this chromosome is close to sequences 10 million base pairs away. And here it's 3 or 4 million, here it's 5 million, here it's 10 million base pairs away. How can every sequence be close to sequences that are 10 million base pairs away? It's a helix. So you have the normal high C map you have, which is things that are close to each other, but if you go between the steps of the helix, then depending on how the helix spirals up, you can be up to 10 million base pairs away. So that told us that the chromatin was organized as loops, but those loops were in some kind of a helix. So just to summarize it for those of you, all of you who didn't read the paper, actually there are a couple of people who read the paper. So this is a typical high C map of an interphase cell where if you've heard of TADs, they're hidden in this line, and these are compartments which are active and inactive chromatin. And so what they saw, what they had seen previously and studied, they had studied mitosis before in Job Decker's lab. They saw that the interphase structure goes away. They saw that. But what they had never seen was that the second diagonal appeared. And the interpretation of it is that chromosomes are organized like a spiral staircase with a loop coming off of the end of every step. Okay? But we had a problem, and we're back to the same problem. These chromosomes are huge. The loops are big. We can't fit. The chromosomes are nowhere near the right size if we do our modeling based on these loops. But we're saved by the two types of condensin. And this is why I think condensin 1 is in the cytoplasm, if there's a reason. It's in the cytoplasm because it evolved to be in the cytoplasm. But if there was a selection for it, this would be one reason why. So during the earliest part of mitosis, the chromosomes, which are red here, are condensing inside the nucleus. And condensin 1 is outside. So condensin 2 is making loops. So it's extruding loops and organizing the chromosome into a chain of loops. So what does that mean when later on here, now the cytoplasm can get in. So condensin 1 wants to make more loops. Oh, I didn't tell you one fact. There's 5 times more condensin 1 than there is condensin 2. So condensin 1 wants to make loops, but it goes in there, and, oh, too late. There are already loops. So it makes loops inside the other loops. So Anton's simulations reveal that there have to be loops within the loops. And so here is a simulation by him with condensin 2 in red and condensin 1 in blue. So prophase, loops are growing. the nuclear envelope breaks down, and the cytoplasm condensin 1 jumps in, and it makes loops inside the loops. Now, what happens when you do that? You end up with a structure like this. So there's a helix, condensin 2, condensin 1, and loops inside loops. And we think it's the loops inside loops that actually compacts the chromosome. Remember, there was the second program that was making things compact. I don't have time to talk about that today. So we started in prophase with loops which eventually become quite large, 400 kb, and then condensin 1 jumps on and it makes loops inside those loops, and then you end up with this compact structure. That was where we were in 2018. So what did we know? Chromosomes are organized as loops by a non-histone scaffold. Sequential binding of condensin 2, which makes big loops, and condensin 1, which jumps inside, organize the chromatin fiber. We call them nested loops. Cohesion is still there, but its function is not known. So another thing that was nice about this, yes, it's a helix. Yes, it looks random. Yes, they're loops. Everybody was right. So nobody was wrong. So that was really great. We liked that. So we then wanted to ask a number of questions. So for example, what do the individual protein complexes actually do when you form chromosomes? And to understand that, we need to know exactly how many copies of all these guys are there. And we needed to know stupid things like, how big is a chromosome? I mean, like, how dense are the nucleosomes? If you're a polymer modeler, how dense are the nucleosomes? So how are we going to do that? So these are our latest studies. We're still writing the paper because you try writing a paper with a bunch of theoretical physicists. Let me tell you, about every 2 minutes, it's like trying to herd cats. They're off in a different direction. So we've been writing this paper for like a year and a half. So we may end up saying some different things, but I hopefully won't say anything that's wrong. So there's still a lot of cohesion there, remember, right? This was 5 times more abundant condensin 1, but there's just as much cohesion as that. So what's it doing? So one of the things that we wanted to know is, what happened? If there's cohesion there, and the cohesion is holding the 2 replicated DNA molecules together, and condensin is trying to make loops, how can condensin make loops if cohesion is there? So one possibility is that when condensin hits cohesion, it stops. I mean, that would make sense, right? And that would give you an organization like this. And Samir Abraham has just recently finished his PhD in Leonid Mirny's lab, and now all these biotech companies in Boston are fighting over him. It's quite nice for him. Here he modeled what would happen if we have condensin hits cohesion and it stalls. So you would end up with a structure. This is 2 sister chromatids, but you would end up with a structure that looks like that. And we know that's not what things look like. So it turned out that the only way you can end up with what looks like chromosomes is that when condensin hits cohesion, it can jump over it somehow and keep extruding the loop. And how you do that topologically, pushing the molecules through the ring, I don't know. It doesn't work like that. In fact, we don't know how it works. But Samir predicted that if that would be the case, his simulations show you can actually separate the 2 sister chromatids, and you end up with 2 sister chromatids separated and cohesion in the middle. So if you did a diagram of it, it would look like this is the blue is the DNA, this is the scaffold, the condensin in each arm, and this is the cohesion in the middle. If you actually look at cells, you're able to see all these molecules, which is extremely difficult to see the cohesion. You can see the condensin on either side and the cohesion in the middle. So this is data. That's a model. That's pretty good, I think. So condensin can jump over cohesion that's holding sisters together, and that's some pretty fancy gymnastics. I don't know how it does that. Now, is cohesion important for the structure of mitotic chromosomes? It's not supposed to be, because it's supposed to be gone. So these are wild-type chromosomes, this is the helix with the spiral staircase in the middle. Let's get rid of condensin 2. That's the one that was acting in interphase and making the big loops. And so we have only the one that makes the nested loops, and they're smaller loops. So we have squiggly-looking chromosomes, long and thin squiggly-looking chromosomes. So if we get rid of condensin 1, which makes the nested loops, we should have only big loops, and so we have short, fat chromosomes. But now let's get rid of cohesin. They don't stay the same. They don't look the same. First of all, you can see we've gotten rid of cohesin because the sister chromatids are separated, but they look really weird. And so we decided to look at what these look like by high C. And the high C, this is another example. The high C blew us away. So this is what we saw. We're used to seeing this. Okay, now let's get rid of the one that makes the little loops. And this high C profile, it's insane. There's a diagonal, there's another diagonal, and there's another diagonal. So these chromosomes have a very bizarre, it's like a helix of helix of helix. It doesn't make sense. And if you get rid of the one that makes, if you get rid of condensin 2, you have no helix, no nothing. That looks like a super simple pattern. These look super complicated. This took Anton 5 times longer to figure out how to model what was going on. And so that's what the chromosomes looked like. So we now have to start thinking about how we're going to model it. So is the scaffold a rod and the loops are coming off of the rod, like the staircase? Is it a solid staircase? But it doesn't have to be a staircase. Maybe it's just things holding loops and they're distributed. So let's actually look at a scaffold protein. So let's go back 30 years or more and look at topoisomerase 2. So this is a chromosome centrifuged onto an electron microscope grid and stained for topoisomerase 2. And we don't see any rod. We see a bunch of spots. Let's blow them up. If we blow them up, those spots are antibodies. See, the black dots are colloidal gold. So each of these spots is a cluster of topoisomerase 2. So the scaffold is a series of spots. It's not a rod, and it's not actually a staircase. Here's another example. So the experiment was really hard to do, but the conclusion is easy. The scaffold, at least of topo 2, is a network. We need to do this for condensin, but we haven't done it yet. We've been doing other things. So we see the central line, the first diagonal we used to see, the second diagonal, and there's a third diagonal. You can see these here. What does it mean? What does it mean? How can we figure it out? Okay, gone on strike again. Yeah. So we needed mathematical modeling to tell us what was telling us. We couldn't figure this out without it. And the wizard is Anton Goloborodko. Samir, I already showed you the modeling. They both learned from Leonid Mirny. That's part of Leonid Mirny there, and that's Anton. And I've been in a Zoom phone call with Anton Goloborodko while he was explaining his models. And what we could see on the screen was computer code, and he was programming in Python while he was talking to us and showing us how to change the features of the models. He's something else. He went from being a PhD student to being a group leader. Right. So and to get the next thing you need a genius microscopist, and that's Fernanda. She's amazing. So what we're going to do is we need to tell Anton exactly what is the density of chromatin in these chromosomes. We didn't know how, there's no easy way to do it. So the way we decided to do it was to use this is a DNA stain called DRAC5, which binds specifically to DNA. And then by adding diaminobenzidine and shining the appropriate light on it, we can convert it to something you can see in the electron microscope. Then we use the same serial sectioning electron microscope, and we can build a model of the cells. And you can see this is being done in an image processing program. It's called Amira. You have to take a month-long course to begin to learn to use Amira. Amira is really complicated. And by the way, it's Allison who runs the microscope and Ian Pryor who got the microscope. And they're in Liverpool, and they're great collaborators. So now what Fernanda's going to do is she's going to build three-dimensional structures of each of these mutant cells. So this is a wild-type cell. If we get rid of cohesin, if we get rid of cohesin and the condensin 1, and if we get rid of cohesin and condensin 2. And so she can see that the volume of these chromosomes is about 70 cubic microns. So we know the size of the chicken genome, so that tells Anton how many cubic microns it is, or how many cubic angstroms it is per nucleosome. These chromosomes have a different size, but that's another issue that we're going to come to later. So then the other thing Fernanda needed to know was what's the diameter of the chromosomes. That was another thing that Anton wanted to know. So using a mirror, she can define the midplane of each of these three-dimensional maps and then measure the diameter. So those are the three different types of chromosomes. No cohesin, no condensin 2. Sorry, the 2 got buried there. No cohesin, no condensin 1. So you can see they're different thicknesses. And you can also see something else. These chromosomes look like what you all think chromosomes are supposed to look like, which is kind of like cylinders. This doesn't. In the absence of, oh my gosh, this is, yes, I'm sorry about this. This is no condensin 2. This is no condensin 1. So this is big loops, lots of tiny loops. And what you can see is that's not a cylinder. In fact, the problem that Anton had was he was doing his modeling and doing his modeling and doing his modeling, and his modeling all involved setting up a cylindrical chromosome and moving the polymer inside it. The only way you could model this was to set up a random coil. It's a random coil. So it's fundamentally different organization. So he used the data, and these are his models. We're still working on how to display these models. These are a week old, these calculations. And so that's no cohesin, that's the chromosome. No cohesin, no condensin 1, big loops by condensin 2, no cohesin, no condensin. All right, so, and a change from our previous thing where we said we thought there was a spiral staircase. No, the models don't work if it's a spiral staircase. The models only work if it's individual spots, like what I showed you for topoisomerase 2, the way it actually is. So the bases of the loops are separated from one another. And so we can start to understand these high C profiles. So let's look at chromosomes that are only expressing condensin 1. So they don't have the big loops. They have little loops. And here we just draw a few loops. And you can see that the loops are far apart from one another. So they never get close enough to one another to have a helix. And so there's no bump on this curve. There's no helical diagonal out there. This is actually, for those polymer physicists in the audience, the slope of minus 1.5 says it's a Rouse polymer, which is a random coil. So this is just a random coil. But remember the high C plot that had all the other lines on it. Well, this is what the modeling shows when you have condensin 2 but no nested loops. The loops are huge. And so the way we think you get the multiple lines is this loop can touch the next loop above it, but also can touch the next loop above that. And that's where the multiple diagonals come from. And what I might point out to you is that this is an experimental curve, and then on top of it is a modeling curve. And I defy you to tell the difference between the two. The model is pretty perfect. So there are distinct roles for condensin 1 and 2. The chromosomes are helical. To some, so another thing that I didn't point out, let me see if I can go back. If you look at this helix, let's go even back. If you look at this helix, it's not a very, if you wanted to draw a model of DNA or something, you wouldn't draw it like this. It's not a very nice helix. It's actually very random. It's just, this is like doing single cell analysis. Each cell, each chromosome has an organization, but it's very statistical. They're very, very variable. They just tend to be helical over time, but they're actually going all over the place. They're not nice, neat helices. So we've drawn that there as a nice helix. They're not nearly so nice as that. If you don't have condensin 2 is responsible for making the helix, and it's responsible for making the chromosome a cylinder instead of just a random coil. So we have long, floppy, random chromosomes. If we don't have condensin 1, the one that jumps on, we have these huge helical loops. The weird thing is the high C profiles look amazing, but the chromosomes look very messy. These chromosomes look really cool. The high C profile looks really boring. These chromosomes look really boring. The high C profile looks really cool. I've got to get your head around that. But the physicists have no problem with that, but the cell biologists are a little disappointed. Okay, so what have we learned? So we've learned that chromosomes are rod-like cylinders, which they consist of nested loops of chromatin, and they're formed by a non-histone structure of proteins composed of SMC proteins. It's not really a structure, it's a network. Interactions between cohesin and condensin are important to form the scaffold. So why is cohesin important? Well, one way to think about it is, so I don't know if the Zoom people can see me or not, but anyway, one way to think about it is I have two sister chromatids side by side. They're held by cohesin. This chromosome wants to coil up nicely. This chromosome wants to coil up nicely. But they're handcuffed together all along their length by cohesin. So this one starts to coil, but then the handcuff won't let it go anymore. So my interpretation of what goes on is that as long as the sister chromatids are tethered together by cohesin, each one can try to make an ideal structure, but it can't do it. If we get rid of the cohesin links, that's why we see more extreme structures, because each chromatid can do what it really wants to do. All right, so that's the role of cohesin in forming chromosomes, is it stops what the two sisters, it makes the two sister chromatids less independent of each other. So chromosomes lacking cohesin and condensin 2, they're no longer rod-like cylinders, they're random coils. Polymer simulations reveal that you can have two chromosomes held together by crosslinks, and somehow or other, this molecule that's making loops can jump across those crosslinks. We don't know how it can do it, but it can do it. And the scaffold, the latest models, and they were hard to see, and we're still working on the model, the image, producing the figures for the paper for that. The scaffold is a chain of SMC complexes, but it follows a helical path, but it's a very disordered helical path. We love order. You know, the DNA is a helix. That's perfect. So, and then we think there's a sort of a helix around the nucleosome. And there was a long time when people thought that the next range of structure was a helix of nucleosomes. We want things to be ordered, but they don't have to be ordered. They just have to be good enough. So polymer simulations reveal there is, in fact, no structure, no single structure for chromosomes. Chromosomes are sort of populations of tendencies. So what don't we know? Well, it's been 140 years since Fleming named mitosis, and we still don't know how the chromatin fiber is organized in chromosomes. We think it's loops. We think the loops make a helix. But if you ask me to be more specific than that, I can't do it. Nobody can. We have atomic structures of these condensin complexes, but we can't tell you how they give the cylindrical shape. We can't tell you how condensin jumps over cohesin. We just don't know. And our polymer models can't explain why it forms a helix. Why does it form a helix? Why isn't it always just a random coil? We don't know. And we don't really know how the loop extrusion works. I showed you the picture which showed the nice convenient little ring with DNA being pushed through it, but you can actually use CRISPR, you can use dCas9 to attach a polystyrene bead, which is huge to the DNA, and it ends up out in that loop. Well, it didn't go through the little ring, so we don't know how it works. So I've been studying chromosomes for over 40 years, and they're still hiding their secrets, and they'll be hiding their secrets after I'm gone, I think. So it's a fascinating system. So what we need new ideas, and we need new techniques, and we need brave people who want to work on this. Okay. So I have this amazing group of people I work with. Kamiko, who is just incredible. System design, genetic constructs, cell lines, high C samples, all that. 3D microscopy by Fernanda. The CHEP experiments done by Itaru. This is Job Decker, who invented C technology, 3C, high C, and all those. Johann actually did it. Leonid Mirny is the polymer physicist who trained both of these guys, Anton, who's in Vienna, and Samir, who's going to be a very rich guy in Boston, not very long from now. And I have to also thank Allison, who ran the microscope, and Ian, who bought the microscope in Liverpool and lets us use it. And the inhibitor we use, you can buy one in MPP1, but I don't know why. If Jim and Lin Fing make it for you, it works better. And they do it for fun. And yes, Ji-Hun already showed, this is the team. Every Wednesday afternoon, except today, we get together, and I can't understand at all what they're talking about. But it's really exciting. It's like if you're a little kid reading a comic book about what science is supposed to be like, you want to have a phone call with Anton, Leonid, and Job. And I thank you for listening, and I thank the Wellcome Trust for supporting us in the way we would like to be supported. Thank you.